reticular activating system and to arousal and awareness. But how does that modulation of cortical excitability and of processing of information by the cortex actually occur? So we know now that that occurs through at least 3 interrelated pathways and mechanisms. The first pathway is the reticulothalamocortical pathway. So we have axons from neurons within the reticular formation that project to the non-specific thalamic nuclei. So the non-specific thalamic nuclei are not relay nuclei like those that project, say, to primary somatosensory cortex. These are non-specific nuclei that project widely through the cortex. So reticular formation neurons project to the centromedian nucleus as well as to parts of the ventral anterior nucleus that act like the non-specific thalamic nuclei. Non-specific thalamic nuclei have very wide and diffuse projections throughout the cortex, with that region of ventral anterior nucleus projecting particularly heavily to frontal lobe, which remember is involved in executive function and regulating the functions of many parts of the cortex. So effectively, this activation of non-specific thalamic nuclei by the reticular formation modulates cortical excitability levels, and it does so in a very widespread fashion throughout much of the cerebral cortex. In addition, we have a reticulothalamothalamocortical pathway. So the neurons in the non-specific thalamic nuclei, like the centromedial nucleus, project to specific thalamic nuclei. So the non-specific thalamic nuclei, like CM, modulate sensory transmission through specific thalamic nuclei, relay nuclei, like the ventral posterior medial and the ventral posterior lateral nuclei of the thalamus. Activation of this reticular formation to thalamus, to other part of the thalamus, to the cortex pathway activates and promotes the transmission of sensory information to the cortex, facilitates activation of the cortex in response to sensory stimulation, and also facilitates cortical processing of that sensory information. I told you earlier that if we have a significant lesion at the level of the upper brain stem, at the level of the midbrain, that can impair arousal, but by interrupting the reticular activating system and produce a coma. It probably shouldn't surprise you at this point then, after having looked at these pathways in the reticular activating system that go through the thalamus, that we also can get impaired arousal with a thalamic lesion. It probably would need to be a bilateral thalamic lesion affecting the thalamus on both sides. But remember that the thalamus, and particularly the key regions of the thalamus involved in the reticular activating system, are located near the midline, and so it can occur that we could get bilateral damage to these key regions of the thalamus and get impaired arousal through interruption of the reticular activating system with a lesion at the level of the thalamus. In addition to these very specific pathways through the thalamus, the reticular system is involved in modulating cortical activity and awareness and processing through a variety of neurochemical systems. So as a group, there are neurochemically defined groups of neurons in the brainstem reticular formation that provide diffuse and modulatory projections really to most of the central nervous system. So they project to the cerebral cortex, to the hypothalamus, to the limbic system, to the basal ganglia, to the thalamus, as we've seen, and to the cerebellum, and to other parts of the brainstem, as well as to the spinal cord. So there are several distinct neurochemical systems coming from relatively defined populations of neurons in the brainstem reticular formation that are involved in modulating cortical activity and processing and awareness. There are cholinergic neurons in the brainstem, particularly in the reticular formation of the pons and the midbrain in a region called the pontomesencephalotegmental complex that project to the thalamus and promote excitability. So these neurons are a key component of the reticulothalamocortical and the reticulothalamothalamocortical systems that we just talked about. These neurons in the pontomesencephalotegmental complex also have other projections to other regions, particularly to basal ganglia and other extrapyramidal motor systems. In addition to those cholinergic neurons in the widely to the cerebral cortex and also play a key role in arousal. Now, these cholinergic basal forebrain neurons in the medial septal nuclei and in the basal nucleus of Maynard are themselves targets of axons from the reticular formation, so they are integrated into another projection pathway by which the ascending projections from the brainstem reticular formation ultimately promote excitability in the cerebral cortex. Now, the primary functions of these cholinergic neurons and their projections, as we said, is to promote excitability, and a variety of studies indicate that they are really very important in facilitating learning and memory functions. There are noradrenergic neurons located in a region of the rostral pons called the locus ceruleus. These neurons, like the cholinergic neurons, have excitatory projections, but they project much more broadly than do the cholinergic neurons that we just discussed. So these noradrenergic neurons in the locus ceruleus have widespread projections through broad regions of the cerebral cortex. They project to the thalamus and the hypothalamus. They project to the cerebellum. They project to other parts of the brainstem, and they project to the spinal cord. So the evidence is that these noradrenergic neurons have critical influences on attention and cortical arousal. They feed into regulation of sleep-wake states. Like the cholinergic neurons, they're involved in facilitating learning and memory, but they also can contribute to regulation of mood and anxiety, and can even influence the speed of information processing by higher order circuits in the cerebral cortex. Populations of neurons that are responsible for most of these brainstem modulatory systems are pretty difficult to locate anatomically. It takes a lot of time and experience, but the neurons in the locus ceruleus are one group that can, with a little effort, be recognized and located within the pons. So we're looking here at a cross-section, an axial view, essentially a cross-section through the pons, and the region of locus ceruleus on one side has been circled in red. So I'll give you here a histological section or hemisection through that region of the pons. So you can see here's the fourth ventricle. So we're just seeing one side of the pons here. And the cluster of neurons that form the locus ceruleus are pretty well defined and visible in this section. So I'll give you just a moment to stare at this and see if you can visually identify that cluster of neurons that is the locus ceruleus, the source of these noradrenergic projections from the upper pons. So this population of neurons in this region has been shaded in green, and this image on the right then represents the locus ceruleus, the location of these noradrenergic neurons on one side of the pons, and then the locus ceruleus on the other side in the corresponding position on the other side of the pons as well. In addition to the cholinergic projection systems and the noradrenergic projection systems, there are dopaminergic neurons located in the ventral tegmental area of the midbrain, not too far from the substantia nigra. These dopaminergic neurons and their projections can be excitatory or inhibitory. Their effects are receptor dependent, so it depends on what specific dopamine receptors are expressed by their targets. But those neurons in the ventral tegmental area have projections to the cortex, but somewhat more limited than those that we looked at in the context of the noradrenergic projections. So these dopaminergic projections from the VTA are primarily to the frontal cortex, particularly to the prefrontal cortex. They also have projections to the nucleus accumbens and to the amygdala that are not indicated in this particular diagram. So in addition to playing a role in regulating cortical processing, these projections seem to be particularly important in influencing the reward pathways and the pleasure systems that you will talk about in later learning events, particularly in the behavioral medicine course. There are important populations of serotonergic neurons that are located along the midline of the reticular formation at the level of the midbrain, the pons, and the medulla. The neurons in what are called these raphe nuclei have very extensive projections, both ascending up the neural axis and descending down into the spinal cord as well. These are primarily inhibitory projections, and those from the upper brainstem are really important, play a major role in regulating the sleep-wake cycle and transition to lower levels of cortical activity associated with sleep. These projections to higher levels of neuroaxis also are involved in regulation of mood and in emotional behavior and aggression as well. The serotonergic projections from the lower part of the brainstem, consistent with our general theme that projections from the lower part of the reticular formation are primarily descending. The serotonergic projections from the lower brainstem play a role in modulating nociception and the transmission of nociceptive information up from the spinal cord, and can also modulate the activity of other populations of cholinergic neurons that are sending from the spinal cord up to higher levels of the neuroaxis. Finally, there are histaminergic neurons that are located in the tuberomammillary nucleus of the posterior hypothalamus. Now you may be thinking, wait a minute, what's this about the hypothalamus? I thought we were talking about the reticular formation of the brainstem. Well, portions of the thalamus and the hypothalamus are functionally considered to be part of the reticular system, even though anatomically they're not located within the reticular formation of the brainstem. So this is one of those important points where we have to differentiate between the reticular system versus the reticular formation, the anatomical organization in the brainstem. These neurons are close to other populations of neurons that produce hypocretin, also called orexin, which you may recall is involved in sleep regulation. And they project to a number of different cholinergic and monoaminergic cell groups to help regulate the transition between wakefulness and sleep. So these play a very specific role in regulating the transition between different states of cortical arousal and wakefulness. They also have additional projections to the midbrain and forebrain that regulate other functions that are beyond the scope of this particular learning event. And if you need to, just remember that many antihistamines actually have sleep-inducing functions consistent with the function of this histaminergic system in the brainstem. So to summarize, these neurochemically defined neuronal populations that are so important for influencing consciousness and processing at the cortical level. We have cholinergic neurons in the tegmentum of the rostral pons and the midbrain that are really critical for arousal. They also are involved in the regulation of REM sleep. We have additional cholinergic neurons, not in the brainstem reticular formation, but rather in the basal forebrain, that also project widely in the cortex and are really important in promoting cortical function, particularly and in promoting attention and memory. We have dopaminergic neurons in the ventral tegmentum of the midbrain that are particularly involved in promoting motor behavior in the context or in response to reward. We have the noradrenergic neurons in the locus ceruleus, particularly important in regulating attention, particularly in response to novel and challenging stimuli. They also are involved in regulation of REM sleep. In this case, particularly, they inhibit REM sleep. We have the serotonergic neurons in the raphe nuclei, also involved in inhibiting REM sleep as well as in regulating mood and affect. And then finally, we have those histaminergic neurons in the tuberomammillary nucleus that are involved in promoting wakefulness. We established before that we can get impairment in arousal with a lesion at the region of the upper brainstem at the midbrain that interrupts the ascending reticular activating system, and we also can see impaired arousal with a bilateral lesion at the level of the thalamus that impairs those pathways in the reticular activating system that go through the thalamus. We also can see impaired arousal, or at least impaired awareness, essentially a vegetative state, with problems at the level of the cerebral cortex. But keep in mind that ultimately all of these projections in the reticular activating system project very, very widespread fashion throughout the cerebral cortex. So to get impairment of awareness due to problems at the level of the cerebral cortex, those problems really have to involve very, very widespread regions of the cerebral cortex, both hemispheres and wide diverse regions of the cerebral cortex being affected at the same time. So this is less likely to occur in the case of, say, a stroke affecting a discrete portion of the cerebral vasculature, more likely to occur in something like a metabolic problem that can occur and affect the function of neurons essentially throughout the entire cerebral cortex. Now let's move out of the rostral brainstem and the rostral components of the reticular formation down into the caudal portion of the reticular system and focus on modulation of motor function by the caudal portions of the reticular system, and we'll spend most of our time looking specifically at the function of the reticulospinal tracts. So our focus for the next few minutes is going to be on the role of the reticular system specifically in modulating voluntary movements. And we'll be looking specifically at the reticulospinal system that plays a major role in modulating the activity of the axial and proximal musculature, particularly in the context of the regulation of posture and in locomotion. Before we look at any detail at the reticulospinal system, I'd like you to watch just a couple of short video clips that demonstrate nicely, I think, important aspects of the activity of those muscles that are involved in locomotion.
So this is what you've seen. So this is me strapped up with EMG electrodes and with reflectors so that I'm filmed, and you can monitor, transform this motion into individual stick figures, and you can see the pattern of muscle activity on the left and right side during locomotion. So this is the alternating pattern that controls every aspect of movement. The horse, like many mammals, many animals, has the ability to alter their locomotor gait from walk to trot to canter to gallop. And this property is not confined to animals like horses. It's also seen in humans. They have the ability to alter gait pattern. I'll show you one demonstration of this. So here on the left, your experimental subject is walking, and on the right is using a different locomotor strategy to get to the same place. And again, you can monitor this, see the individual limb movement, see the alternating pattern of muscle activity on the left, the co-activation on the right. So interneurons are the final mediators of this, and presumably their response.
Locomotion is a voluntary motor activity, but it requires a tremendous amount of unconscious neural processing. Walking and running requires rhythmic movements that involve all four extremities, and normal walking or running requires reciprocal activity of the extremities on each side and also reciprocal activation of extensors and flexors. But as shown in the last video, the normal pattern of activity can be altered under some conditions. Coordinating and executing this muscle activity requires the involvement of several interconnected neural systems. In humans, locomotion is actually initiated from a locomotor center that's located in the lower midbrain. The basic locomotor patterns are intrinsic, but they're modulated by inputs from higher neural centers coming from the cerebral cortex, and also modulated by sensory feedback. The premotor cortex has direct projections to the brainstem upper motor neurons that are the source of the reticulospinal tracts. And the reticulospinal tracts modulate and, when necessary, override the motor activity generators that control locomotor activity. Two reticulospinal tracts originate on either side of the brainstem, and they descend and act via interneurons to regulate the activity of lower motor neurons that control the axial and proximal limb muscles. And they share this control with the corticospinal fibers. The medial reticulospinal tract, shown in red in the diagram, originates in the pontine reticular formation, and the axons descend ipsilaterally in the anterior white matter of the spinal cord to regulate axial and extensor motor neuron. The lateral reticulospinal tract, shown in blue in the diagram, arises in the medullary reticular formation and descends partly crossed in the more lateral